Written everyone, psoriasis, definition and epidemiology. Introduction. Psoriasis is an immune-mediated polygenic skin disorder. Various environmental triggering factors, trauma, infection, medication may elicit disease in predisposed individuals. The characteristic lesion is sharply demarcated erythematous plaque with Mercator scale, and the plaques may be localized or widespread in distribution. Histological hyperkeratosis, parakeratosis, acanthosis of the epidermis, tortuous and dilated vessels, and inflammatory infiltrate composed predominantly of lymphocytes are observed. Psoriasis is a systemic disease process in which up to 20 to 30 of patients have and will develop psoriatic arthritis. In addition, in patients with moderate to severe psoriasis, there is an increased relative risk for metabolic syndrome, atherosclerotic, cardiovascular disease, and depression. Psoriasis also has a significant impact on patient quality of life, and in survey, patients feel that the current treatment, although often effective, do not provide a satisfactory long-term solution. History. Hippocrates and his school provide meticulous description of many skin disorders in their classification. Tri-scaly eruptions were grouped together under heading low point. This group properly includes psoriasis and leprosy. Between 129 and 99 BC, the word spora, meaning desquamated condition, was first used by Galen to describe a skin disorder characterized by scaliness of the eyelid, corner of the eyes, and scrotum. The condition was sclerotic and excoriation would resent on those called psoriasis. This affliction was rapidly the type eczema. It was not until the 19th century that psoriasis was recognized as an entity distinct from leprosy, although Robert Wilson was first to give an accurate description of psoriasis. It would be another 30 years before Hebra definitively separated the clinical features of psoriasis from those of leprosy. In 1899, Heinrich Klopner described the development of psoriatic plaques as sign of injured skin. The World Health Organization passed a resolution in 2014 recognizing psoriasis as a chronic, non-communicable, painful, disfiguring, and disabling disease for which there is no cure. Epidemiology and genetics. In most reviews, the prevalence of psoriasis is said to be 2% of the world population. However, in the US and Canada, prevalence is as high as 4.6 and 4.7 have been reported, respectively. This contrasts with frequencies in African American, Norwegian Lab, or Asians of between 0.4 and 0.7%. A worldwide systematic literature review was performed by the Global Psoriasis Atlas, focusing on the epidemiology of psoriasis. The incidence varied from 30 per 100,000 person years in Taiwan to 321 per 100,000 person years in Italy. Psoriasis proved to be more frequent in high-income countries and in regions with older population. One population-based study noted that 73% of affected individuals had mild to moderate psoriasis, while 27% suffered from more severe involvement in one large group of patients with psoriasis. 79% of nail changes, psoriatic arthritis, has been found to affect 5% to 30% of patients with cutaneous psoriasis, depending on the series. Psoriasis can first appear at any age, from infancy to 8 decades of life. Two peaks in age of onset have been reported, one at 20 to 30 years of age, and second peak at 50 to 60 years. In 75% of patients, the onset is before the age of 40 years, and in 35% to 50%, it is before the age of 20 years, although the age of onset is earlier in women than in men. The natural history is similar, chronic with intermittent remission. In one epidemiology study, 39% of the patients stated they had experienced remission of 1 to 54 years. In Europe, the overall prevalence rate for juvenile psoriasis was found to be 0.7%, with an increase from 0.37 to 0.55% in those from 0 to 9 years of age, to 1.01 to 1.37% in those 10 to 19 years of age. Plaque psoriasis is the most frequent form of the disease in children, followed by cutaneous psoriasis. Genetic factor. Depending upon the series, the positive family history has been reported by 35 to 90% of patients with psoriasis. Based on last survey-based study in Germany, in both parents had psoriasis, the risk of their child developing psoriasis were 41%, whereas if only one parent were affected, the risk were 14%, the risk was 6% if just one sibling had psoriasis. Analysis of concordant rates among monozygotic and dizygotic twins is another method for examining the influence of genetic factors on the disease. One literature review found that 141 monozygotic twin pairs, 82 were concordant with psoriasis, and 59 were discordant. Of 155 dizygotic twin pairs, only 31 were concordant and 124 were discordant for psoriasis. Thus, there is a 2 to 3-fold increased risk of psoriasis in monozygotic twins as compared to dizygotic twins, implying that genetic factors are important. The distribution of the lesions, the severity, and the age of onset was similar in the monozygotic twin pair, whereas their features differ in dizygotic twin pairs. This observation suggested that genetic factors also play a role in the clinical course of psoriasis. HLA studies. Histocompatibility antigens. HLA are surface antigens on human cells, and the corresponding chromosomal region is called the major histocompatibility complex, MHC. It is situated on the short arm of chromosome 6. Psoriasis is associated with HLA-CW6 with the presence of HLA-CW6, conferring a relative risk of 13 for developing psoriasis in the white population and 25 in the Japanese. HLA-CW6 is strongly linked to the age of onset of psoriasis. In one series, HLA-CWC was expressed in 90% of the patients with early onset psoriasis, in 50% of those with late onset psoriasis, and only in 7% of the control population. A specific MHC class II antigen also appeared to be associated with the early onset psoriasis, and the psoriasis-associated HLA alleles were often in an extended haplotype. CWC B57, DRB1, O701, DQA1, O201, DQB1, O303. Individuals carrying this haplotype were found to have a 26-fold increased risk of developing early onset psoriasis. As a result, some clinicians have designated patients with early onset psoriasis a positive family history of psoriasis on expression of HLA-CWC as having type 1 psoriasis, and those with late onset disease, no family history, and the lack of expression of HLA-CWC as having type 2 psoriasis, while HLA-CWC will serve as biomarker for clinical and therapeutic stratification. Today, there are no treatments that specifically target HLA-CWC or the HLA alleles can be associated with different psoriasis variants and related conditions. For example, the HLA-B27 allele is a marker for sacroiliitis-associated psoriasis and reactive arthritis. Classic genome-wide linkage analysis identified at least 9 psoriasis susceptibility regions, SOS1 to 9, in different chromosomal locations. By far the most important genetic regions were SOS1 on chromosome 6p, which is estimated to account for up to 50% of psoriasis risk. SOS1 contains genes such as HLA-C with the HLA-CWC allele and corneodesmosine. Due to high linkage disequilibrium in SOS1 genes within this region and inherited as a block, it has been challenging to determine which genes within SOS1 contribute to psoriasis pathogenesis. In genome-wide associated study, in which hundreds of thousands of single nucleotide polymorphisms across entire human genome are examined in thousands of patients, at least 80 risk loci have been identified. It has been estimated that this loci explain 28% of genetic heritability. GWAS have led to several conclusions regarding the genetic factor in psoriasis. First, most of the genes that have been implicated had immune-related function, underscoring the importance of the innate and adaptive immune system in the pathogenesis of psoriasis. In contrast, relative few genes that encode skin-specific protein have been associated with psoriasis. Second, thus far surprisingly, few interactions among the genetic variants have been identified, with the exception of HLA-CWC and ERF1. Third, associated genes encoded protein with roles in particular immunologic and signaling pathway, especially those involving tumor necrosis factor TNF, NF-κB, interferon IFN, and interleukin IL-23 Th17 cells. Lastly, ARAP1, which encodes an aminopeptidase involved in MSC class I antigen processing synergistically with the HLA-CWC risk allele, providing another argument for the role of an MHC-restricted antigen and its presentation through HLA-C in the pathogenesis of psoriasis. More recently, greater insight into the genetics of pustular psoriasis have been made in genetics with generalized pustular psoriasis. Several groups identify homozygous or compound heterozygous loss of function mutation in IL-36RN, which encode the IL-36 receptor antagonist. Patients with the autoinflammatory disorder deficiency of IL-36 receptor antagonist presented with widespread psoriasis form plaques, though edges contain sterile pustule. Based upon susceptibility loci and transcriptomics, a significant IL-36 signature was also observed in patients with plaque psoriasis. Functional genomic studies. Microarray technology has been utilized to obtain a comprehensive picture of the genes expressed in psoriasis skin. More than 1,300 genes were found to be differentially expressed when compared to normal human skin. This group of genes include no marker of psoriasis in the skin, but it also contains numerous genes not known to be expressed in the skin. The analysis confirmed a genomic scale, the involvement of T cell and dendritic cell (DC), and also indicated for a role for comorbidity associated genes involved in the atherosclerosis, signaling and fatty acid metabolism. Another important gene family differentially expressed in psoriatic epidermis encode antimicrobial peptide. The peptides are expressed at high level in psoriatic skin, but at low level in skin affected by atopic dermatitis. Additionally, complete functional impairment of the IL-36 receptor antagonist secondary to known mutation in IL-36RN has highlighted the importance of resurrecting IL-36 dependent activation of NF-κB pathway. Such insight led to the recent introduction of an interleukin-36 receptor inhibitor for the treatment of generalized pustular psoriasis.